Okay, I think exam one will be next week and not the end of this week. I'm going to finish chapter four. I'm going to start chapter five. I won't finish chapter five today because I have another meeting to go to, and I'll have to leave to go to that meeting a little after two. And it's going to be a short day. I have posted some homework on Canvas. It's in the form of a bunch of dihybrid crosses. There are six problems on that handout. You only have to do three. And I'm only going to look at the three you do correct. And I'll give you 2 points per. So that's six total points. And I have some more homework on chapter three on Connect. And I will put some more on chapter four and chapter five online before the end of this week. And so this is very briefly the end of chapter four. I almost got done with it last week. But here it goes, sex determination and the different types of sex determination. And then we very rapidly go into mammalian model, and it's accompanied by the fruit fly model, which in this presentation is essentially basically the same thing. In the early 1900s, there was a scientist named Thomas Hunt Morgan, and he was working up, I want to say, at Cornell University in New York. And he was working with fruit flies. He was working with Drosophila melanogaster. And he would either induce mutations in Drosophila, or he would go out into wild populations of fruit flies and look for mutations. And either way, he acquired a fruit fly with white eyes. And he was very clever. He called the new mutation white. And he crossed this white-eyed fly with normal red-eyed flies to see what the inheritance pattern was. And so he did the cross. And it turned out, by the way, that this white-eyed fly he had either made or found or whatever was male. And so he crossed it with a red-eyed female, and he got the F1 generation. And all of the white-eyed flies vanished. And so that was evidence that this white-eyed allele was recessive. And so then he took the two individuals from the F1 generation and he crossed them together. And the white-eyed individual indeed reappeared. And it was a 3-to-1 ratio. And again, that looks like Mendelian results for a recessive allele. But then he did a little further in his work. And he took those white-eyed males and he crossed it to a female from the F1 generation that had red eyes. And in some rather surprising results, he found out that these white-eyed individuals came back, and you would expect them to be in a 1-to-1-to-1-to-1 ratio, and they weren't quite 1-to-1-to-1-to-1. But in this experiment, he had white-eyed males and white-eyed females. And so that implied that the inheritance pattern for this allele was a little curious. And there's his actual numbers crossing the white-eyed males, the F1 females down here, and he gets about a 1-to-1 red-eyed males and females, and a little less than that white-eyed males and females. And this is explained. These numbers are explained in a couple more slides. And so anyway, it was fairly easy to conclude that that particular white-eyed allele is recessive, and the red allele, which is wild type, is dominant. And if you just do those F1 crosses to make the F2 generation, well, you get a very typical Mendelian dominance recessiveness kind of inheritance pattern. 3 red to 1 white. An easy way to look at that is that the white-eyed individuals should be about 50 female and 50% male. And they're not. And so in this particular cross, the only white-eyed flies were male. And if that allele, if that gene was on autosome, that shouldn't be the case. But by this time, it was known the sex determination patterns used in fruit flies. And it was known that the female fruit flies were XX and the male fruit flies were XY. And that would lead to the conclusion that not only was this white-eyed allele recessive, it was on the X chromosome. And that would lead you to this expectation that in this particular cross, all the white-eyed flies should be male. And no females should have white eyes. And so that explained his data very nicely. Now, if you do the math, you don't really get a perfect 3-to-1 ratio of red-eyed individuals and white-eyed individuals. You get approximately a 4.5-to-1 ratio. And this is indicative. This seems to indicate that I'm not saying the white-eyed allele was fatal. I'm not saying that those flies were blind and therefore they couldn't see to get prey or anything like that. But it is indicative that something in those white-eyed flies lowers their ratio of survival. And you can do some other cross. You can do this test cross. And in this particular type of test cross, you're going to take a white-eyed male with a heterozygous female, and you do the cross. And it should allow you to make a prediction that you would expect approximately half the population that is made to be red-eyed and approximately half to be white-eyed. And about half of those white-eyed flies should be male and half should be female. And you can get the real numbers. And the real numbers are about 130 to 130 to about 89 to 89, which would come out to be 1 to 1 to 1 to 1. But the real data is about a 1.5 to 1.5 to 1 to 1. And the 1.5s are red-eyed, and the 1s are the white-eyed. And so that seems to indicate that those white-eyed flies somehow have a lower survival rate. And we think that it's the lack of pigmentation in the eyes that are allowing a greater wavelength of light into the eye and into the fly's brain. And it causes a dazzling effect. It doesn't make them blind. It just makes them observe a little bit more radiation than they should be. Now, you can analyze pedigrees if you really want to trace down that a genetic disorder is on the X chromosome. Best way to do it is through analyzing the pedigree. And there is a type of muscular dystrophy. And it turns out that this gene is on the X chromosome. And it turns out that this gene encodes the protein called dystrophin, which you need in proper muscle structure. And among other things, this mutation damages your heart and your breathing muscles. And eventually you will die somewhere in your 30s. And this disease has been studied for pedigrees, and a few things are common, and especially an X-linked recessive disorder. If you look at the pedigree as a whole, the disease is very rare in females. It doesn't mean it never exists. It just means it's rare. However, the females might be carriers, and they themselves will show no symptoms of the disease. And furthermore, if the mother is a carrier, she will have about 50% of all her sons will have this type of muscular dystrophy. And so you can construct a pedigree. And again, these solid shapes are easy to determine. They have the disease. It's noticeable. You can detect it. These shapes here are all female. They're all halfway colored in. That is indicative of a heterozygote. But that is a guess. Because it could be that in this particular generation, they were just unlucky. And new mutations kept cropping up in the egg cells that this one made over and over and over again. But that is not very likely. And so you assume that she has to be a carrier. He will be affected with the muscular dystrophy or not. And so for the father, it's easy. He has it or he doesn't. For the mother, it's a little bit of guesswork. Now, you can also do what is called a reciprocal cross. And a reciprocal cross is basically, it's genetically speaking, the exact same cross. But in a reciprocal cross, the sex that carries the trait will be different. So it will either be the male or the female parent. And because of the differences in numbers of X chromosomes, the X-linked genes will behave differently if you get that disease from your mother or your father. And anyway, pedigree analysis, reciprocal crosses, and some other things have determined that this form of muscular dystrophy is inherited the same way in dogs as in humans. And there is a dog with the X-linked muscular dystrophy. And here is the reciprocal cross. Unaffected female, affected male. All offspring will not have muscular dystrophy, but all of the daughters produced will be carriers. And do it backwards. Affected female, unaffected male. And you get 50% females that are unaffected, but are all carriers. And you get all the males have muscular dystrophy, because they must inherit that X chromosome with the mutation from the mother. That's the end of chapter 4. Let me stop this, make sure I am recording, and I am. I remember to do that. On to chapter 5. And chapter 5 is basically extensions of Mendelian inheritance. And depending upon when you took Biology 1033 and who you took it from, this is more of a review. It depends upon how much genetics your Biology 1 teacher knew. And so one of the deviations from Mendel is that multiple alleles contribute to a trait. And so in simple Mendelian genetics, and it is very simple, there is one gene with two alleles, and the extent of that trait is 100% determined by the genetics. And so Mendelian, especially monohybrid crosses, easy to analyze. But there are all kinds of variations. And so, again, in some simple Mendelian inheritance, there's two laws: law of segregation and the law of independent assortment. And if simple Mendelian inheritance is being followed, the law of segregation and the law of independent assortment will always be observed. However, simple Mendelian inheritance is the exception rather than the rule. And so most genes have much more complex and interesting patterns of inheritance. And there are some simple varieties. And so there is simple Mendelian inheritance. One gene, two alleles, one is dominant, one recessive. Underlying explanation: the dominant allele encodes a functional protein, and half of the dosage is enough. And then you have X-linked. You have a gene on a sex chromosome. And so males and females have different numbers of these chromosomes. And in some cases, those alleles are simply dominant recessive. And in some cases, 50% of the functional protein is enough. In some cases, 50% of that protein is not enough. And then you have what is called incomplete penetrance. In Mendelian genetics, all the penetrance was 100%. So anything that contained at least one dominant allele expressed the dominant phenotype. In incomplete penetrance, individuals might have the dominant allele, but they don't show the dominant phenotype. And there are several possible underlying explanations of this. And then you have incomplete dominance. This is the famous AB of blood typing in humans. And in incomplete dominance, the heterozygote has a different phenotype than either homozygote. And it's usually more or less the average. Red-flowered plants and white-flowered plants are homozygous. They make a heterozygous that has pink flowers. And the explanation is, well, 50% of the proteins are active, 50% are not, and it causes an intermediate phenotype to be observed. And then you have an example of overdominance, which is kind of unusual. And overdominance, the heterozygote has a phenotypic traits that offer more reproductive success than either homozygote has. And there's at least three possibilities that can explain that. And they get discussed in a little bit. And then you have codominance. Sorry, that was the ABO blood type. And so in humans at ABO blood typing, you could have alleles I superscript B, little I, and the A and the B is dominant to the little I. And so if you have both the A allele and the B allele, you have AB blood type. And essentially you have both blood types A and B. And then you have sex influenced inheritance. Easy explanation is that there are different sex hormones in different individuals of different species of members of species. And it's these sex hormones that can influence the expression of genes. And then you have sex limited inheritance. The trait is only going to occur in one of the two sexes. And again, it can be related to differences in sex hormones. And then you have alleles that are lethal. You have a lethal allele, it causes death. And some alleles cause death during development before birth. And some alleles are what they call conditionally lethal. They're only lethal in certain environmental conditions. And so you want to understand Mendelian inheritance for a couple of reasons. You want to be able to predict outcome of crosses. And you want to be able to understand how gene expression can account for phenotype. And so usually populations are going to have some wild type allele, which is the most common allele found in the population. And they're probably going to have polymorphisms, more than one allele at that gene. And that's the most likely explanation for alleles being dominant and recessive. And to further complicate things, certain things can have incomplete penetrance, and they can vary in expressivity. For example, polydactyly is a condition in which you have more than 5 fingers on a hand. And usually you have, instead of 5, you have 6. But it can be overexpressed, and you can have 7, or you can have 8, or whatever. That's a change in the expressiveness. So wild type allele is the most common version of allele in a population. And usually we say wild type alleles make normal proteins. And they are there to further the reproductive success of the organism. In populations that are big enough, there might be more than one common allele. And this is just polymorphism. And so you can have flowers and plants of the same species that are of different color. And anything that is less common is considered to be some form of mutation. And mutations are rare. And so mutant alleles themselves are rare. And most mutations are knockout mutations. They make recessive alleles. And they usually contain something that reduces the making of some functional protein. And so there is usually some kind of error in the sequence of the gene that causes some kind of error in the amino acid sequence of polypeptides, which usually causes these polypeptides to misfold and make a poorly functional or non-functional protein. There are lots of human genetic diseases that are tied to a single gene, a single allele. And most of those disease-causing alleles are recessive. And there is some of them. And these are all recessive. And these can all have some negative ability on the fitness of the individual that has it. Some of them are relatively minor, and some of them are, well, you have Tay Sachs disease. Early death means usually by about the age of 5 or so. And so some are like really minor. If you're albino, you have certain lifestyle changes. And some of these things are lethal. Now, most mutants are recessive. You have 2 copies of every gene. You have 2 chromosomes, and on every chromosome there is a gene. And a gene is going to be called recessive if the heterozygote is normal. And so that means only 1 wild type copy is enough. And there is at least 2 explanations. And one of them is very simple. Half of the normal levels of functioning protein are enough. The other explanation, since you have 1 wild type gene and 1 mutated one, you upregulate the wild type copy. And so you make more than 50% of the levels of the protein. And so you overexpress the quote unquote good copy that you do have. And here are, some examples where 50% is good enough. You have purple flowers and white flowers, and so you can indicate them with a allele named P or little p. If you have homozygous dominant, you make all of the functioning enzyme that you're supposed to make, and your flowers turn purple. If you're heterozygous, you make 50% of the functioning enzyme that you're supposed to make, but that's enough. And if you're homozygous recessive, you make none. And that means your flowers stay white. Now, some mutations are dominant. And they're much more rare, but it is not unheard of. And there's at least 3 types. Gain of function is the easiest to explain. And then more difficult, dominant negative mutations, and something called haploinsufficiency. In a gain of function mutation, the mutation has caused the protein that is made to have a new or abnormal function. And it might be that it's overexpressed and overproduces itself, or whatever. It might be that you just randomly, through some random discovery, have found out there is some sequence of amino acids that is overactive compared to the wild type. And then you have what is called a dominant negative mutation. The mutated protein that is made acts against the wild type protein. And then haploinsufficiency. Well, the mutant is actually loss of function, and one wild type copy of the gene is not enough. And then you have incomplete penetrance. It's indicative that the allele doesn't always penetrate into the phenotype of the individual that has it. And you generally describe it at the level of the population. And so they say if 60% of the heterozygotes carrying a dominant allele exhibit the trait, then that trait is 60% penetrant. In any given individual, the trait will be there or not. Now, again, polydactyly is due to a dominant allele on one of the autosomes, and it causes more digits on a hand or foot. And so, usually, if you're heterozygous for that gene, you have more than 5 fingers or toes on a hand or foot. However, there are some individuals that are heterozygous for that condition that are normal. And there's a typical pedigree for a dominant trait. It goes, it's found in every generation. And then you have expressivity. If you have one extra toe or one extra finger, you have a low expressivity of that polydactyly trait. If you have several extra, you have high expressivity. And there are 2 possible modifications. The environment plays an effect on your phenotype. And so your phenotype is not 100% determined by your genotype. The environment in which you develop plays a role. And there might be what they call modifier genes. And most of these genes are probably unknown. And these modifier genes are other genes that get expressed and could, modify the trait. And they can be very different in different individuals. Now, the environment has a definite effect on gene expression. And I really hate the term norm of reaction, but Brooker apparently loves it. And it's really easy to explain if you consider, for example, human height. You do most of your growing while you are growing up. And by the time you're 20-ish or so, you're probably not going to increase in height anymore. And during those years in which you are growing, especially while you're a child or a teenager, and your diet is sufficient, especially calories you get from proteins, you might exceed your genetic prediction for height. If, however, during those years that you were growing, your diet is insufficient, you will not meet the genetic prediction that you will have. Arctic foxes and a lot of other organisms change their coat color due to changes in temperature. And so in this Arctic fox, they tend to be brownish in the summer, and when it's cold in the winter, they turn white. If you have phenylketonuria, you can't metabolize phenylalanine. And so that amino acid builds up in your body, and it impairs development of the central nervous system. At birth in America, every newborn is tested to see if they have phenylketonuria. And if that test comes back positive, they pretty much instantly go on a very low phenylalanine diet. And so that amino acid doesn't build up in their body. And so they can develop normally. And so there's the fox. Same fox. And it will just change color depending on the seasons. Someone with PKU that was tested positive at a young age and avoided phenylalanine in their diet. And so they say, norm of the reaction. The range of phenotypes seen due to environmental effects. And there's a variety of ways to figure this out. And so you need to start with a true breeding strain of organisms. And you have to breed them. And you have to raise them in different conditions. And for example, fruit flies have multifaceted eyes. And so they have all these little islets that make up the compound eye of the animal. And it turns out that the number of little islets that a fruit fly clusters together to make an eye depends upon the temperature. And as it gets warmer, as the flies are being incubated, the number of facets goes down. And so you can try to predict outcomes with incomplete dominance, overdominance, or codominance. And we try to explain at the molecular level what's going on in incomplete dominance, overdominance, and codominance. In incomplete dominance, the heterozygote exhibits a phenotype that is intermediate between the two homozygotes that produced it. Example, flower color in certain plants. There are two alleles. C superscript R, which makes red flowers, and C superscript W, which makes white flowers. And notice both of these are capital letters. That indicates that they are not dominant recessive to each other. So you have a red flower and a white flower, breed them together. It has to make a heterozygous individual. It has pink flowers. Cross individuals of that generation together, and you get a chance of observing the two homozygous individuals again and the heterozygous individuals. And so the easiest explanation is because whatever protein this CR allele encodes, half of it is not enough to completely turn those flowers red. Now, it turns out that most traits, when you look at them very closely, they are exhibiting some sort of incomplete dominance inheritance. And when Mendel, for example, looked at P shape, round or wrinkled, just with his eyes, he decided peas could be round or they could be wrinkled. And he came to the conclusion that homozygous are, or heterozygous, made round peas, and the homozygous recessive made wrinkled peas. And therefore, capital R was dominant over little r. But if you look close enough, what you will see is that there is a difference between big R, and the heterozygote. And it makes a difference in the amount of starch that is made. And 50% of the starch that is made is enough to give the pea a round shape according to your naked eye. But if you look at it molecularly and you test for the presence of starch, you will see that there is a difference. And then if you're wrinkled, well, you have nothing. And so you're very low starch. Overdominance is when a heterozygote has greater reproductive success than either homozygote. Also can be referred to as heterozygote advantage. And classic example, your red blood cells are loaded up with a protein called hemoglobin. And hemoglobin is what allows your red blood cells to carry oxygen around. And the normal allele is HbA. And the mutant allele is HbS. Now, if you are homozygous for HbS, your red blood cells sickle. And the sickling is not good for your red blood cells. Assuming that sickle shape on the right is not good for them. And so you get anemia. The sickled cells also now have a long axis and a short axis. And if they turn along their long axis, they can get stuck in the capillaries. And they can do things like cause great pain in certain joints and certain organs. And fully affected individuals with sickle cell anemia tend to have a shorter lifespan than those that are unaffected. Now, it turns out that if certain kinds of malaria are present in the environment, being heterozygous for sickle cell anemia is advantageous. And malaria is caused by a protozoan. And protozoans often have very weird life cycles. And this particular genus goes through one stage in a type of mosquito, and then it gets into your body, and eventually goes through another stage inside your red blood cells. And if you are heterozygous for sickle cell, HbA, HbS, when this plasmodium gets into those sickle cells, when they sickle, they rupture. And when they rupture, they kill that protozoan. And therefore, if you're heterozygous at this allele, you are at an advantage. You do not have full-blown sickle cell anemia, so that's good. And you're more resistant to malarial infection. So that's good. If the malarial parasite was removed from the environment, this heterozygous individual would cease to be at an advantage. And then let's see. I'll stop when I get to codominance. There are some possible explanations of why overdominance exists. At the level of molecule, overdominance can be due to two alleles that make very slightly different copies of the protein. And very often, proteins have to interact with each other to make a functioning protein. And so there are three potential explanations for overdominance. One is increased disease resistance. One is the composition of the proteins that they make. And one is because these proteins exist in dimers or tetramers or something. These slightly different versions of the protein can cause different functions in the dimer or the tetramer or the functional level of the protein. And for example, you have potential alleles A1 and A2. If you're homozygous, for whatever reason, the disease causing agent is more likely to be able to infect your cells. For example, sickle cell anemia and malaria. The heterozygote has at least some resistance. There is another disorder, Tay-Sachs. You don't want to have Tay-Sachs. But there is some speculation that if you're a heterozygous for the gene that causes Tay-Sachs, you're more resistant to tuberculosis. If you're heterozygous for PKU, you could be more resistant to certain types of miscarriage. Some proteins function as dimers. Two subunits have to bind together to make a functioning protein. And so if you have the genotypes A1A1, you make all A1A1 homodimers. If you're homozygous for A2, you make nothing but A2A2 homodimers. If you're heterozygous, you can make A1A1, A2A2, or A1A2. And in some combinations, it's that mix, that A1A2 dimer, that is going to have better activity. And for example, there is a gene. It's given E. You have an allele E1. And E1 happens to function best at a temperature range between 27 and 32 degrees. And you have the allele E2, and it makes an enzyme that functions best between 30 and 37 degrees. If you're heterozygote, you make both enzymes. And therefore, your optimal range for being incubated ranges from 27 degrees centigrade to 37 degrees centigrade. And I'll start there on Thursday, and I better finish chapter 5 by end of Thursday.